Namaste everyone. Happy learning. Welcome to my channel, Pratibha Ashok Kumar from KLSNC. Today I'll be presenting a lecture talk on factors influencing enzyme activity. Okay, so let us go through the factors that influence the enzyme activity or rate of the reaction. First one, substrate concentration, enzyme concentration, product concentration, temperature, pH, time, activators and inhibitors. Okay, see, these are some of the factors that influence the enzyme activity. Even sometimes they may ask you the factors that affect the enzyme activity. You can write the same answer. Now here there is a question: what are the factors influence the enzyme activity or enzyme catalyzed reaction? We can list like enzyme concentration, substrate concentration, effect of temperature, effect of pH, effect of product concentration, effect of activators. Okay, this question will be asked for 2 marks. Further, we can see here enzyme plus substrate which is going to react together, okay, in the active sites of the enzyme, this particular substrate and becomes enzyme substrate complex in turn, which gives the product. Okay, so now let us go through this question. You will get from this particular unit. The question is like this: explain the factors influence the enzyme activity or enzyme catalyzed reaction. Okay, in that now let us go through the first one, enzyme concentration. Okay, the rate of enzyme catalyzed reaction depends on various factors. Now let us go through the enzyme concentration. The rate of enzyme catalyzed reaction is directly proportional to the enzyme concentration. Okay, greater the concentration of enzyme, the faster will be the reaction. At a constant temperature, pH, substrate concentration, the increase in the concentration of the enzyme increases the initial velocity of the reaction. This is due to the fact that large number of the substrate can interact with the enzyme molecules to form product. Okay. Further, as the concentration of the enzyme is increased, the velocity of the reaction proportionately increases. Okay. In fact, this property of enzyme is made use in determining the serum enzymes for the diagnosis of diseases. By using a known volume of the serum and keeping all the other factors like substrate, pH, temperature, etc. at the optimum level, the enzyme could be assayed in the laboratory. Okay, effect of enzyme concentration, that factors that influence, you can write this one or you can also write this information. Okay, hope you know the definition of the enzyme, right? What are enzymes? Enzymes are biocatalysts, accelerate the rate of reaction or increases the rate of reaction or velocity of the reaction without undergoing change. Okay, now let us go through here effect of substrate concentration. Okay, here it increase in the substrate concentration gradually increases the velocity of enzyme reaction within the limited range of substrate levels. Okay, a rectangular hyperbola is obtained when velocity is plotted against the substrate concentration. Okay, that time we will find three distinct phases, okay, of the reaction are observed in the graph. That is A, this A which is the linear, whereas B which is the curve, and C is the almost unchanged. Okay, whereas V is equal to velocity of reaction, then V max, this one V max is equal to maximum rate achieved by the system. Okay, S is the substrate concentration, whereas Km is the Michaelis constant. Initially, rate increases with substrate concentration, okay, and there will be high chances of collision to form enzyme substrate complex and product. Okay, after certain value, rate remain constant. Why? Because of the active site of the enzyme is filled with the substrate and attained saturated condition. Okay, it means when the saturation has occurred, new added substrate are unable to react with active site and becomes constant. Okay, here you can see one other graphical representation. This is substrate concentration. Here, rate of reaction, that is velocity of the enzyme. Okay, here when the substrate concentration increases and reaches the saturation condition. Increasing concentration of the substrate does not affect the reaction rate because it has already attained the optimal condition. The variation in the rate when the concentration of substrate is increased as in the graph. Okay, when the concentration of the substrate alone is increased, keeping the enzyme concentration, temperature, pH constant, hyperbolic curve is obtained as in graph. Okay, this indicates that low substrate concentration, okay, then the rate of reaction increases with increase in the substrate concentration and reaches a maximum condition called, that is called V max, that is maximum velocity. At high substrate concentration, all the enzyme molecules are saturated, hence the rate attains a maximum value and does not alter further. Okay, for this effect of substrate concentration, you can write this particular PPT, or you can write information, or you can write this information. Okay, can see here once they attain the optimum condition, they start, they'll be in the constant. Okay, further there won't be any kind of the reaction. Now let us go through effect of temperature. Generally, the increase in temperature increases the rate of reaction. Since more the reactants gain more kinetic energy, which leads to more number of effective molecular collisions. In enzyme catalyzed reactions also, increase in temperature, see 10, 20, 30, 40 like that, okay? Increase in temperature increases the rate of certain extent, then the rate decreases as in the graphical representation. When this graph decreases when after the attainment of the optimal temperature, okay? And once uh, there the increase in the temperature, that time what happens, automatically there will be a decrease, curve can be observed. At high temperature, the rate of enzyme catalyzed reaction decreases because at the high temperature the enzymes lose their tertiary structure and they get denatured. Okay, because the most of the enzymes which surviving that is in a particular temperature, if the temperature exceeds, they may undergo denaturation where they will lose their native configuration or three-dimensional configuration. The temperature at which the rate of enzyme activity is highest or maximum is called optimum temperature, or OT. Okay, I'll repeat. The temperature at which the rate of enzyme activity highest or maximum called optimum temperature. Okay, for most of the mammalian enzyme, the optimum temperature is 37 degree Celsius. Okay, at low temperature, enzyme activity is nearly zero. Okay, kinetic energy is very slow for enzyme and substrate and show slow movements and no collision rate will be zero. As temperature increases here, when we increase the temperature here, automatically you can see the graph. As temperature increases, kinetic energy increases and more collision increases and more is the rate of reaction. Okay, the temperature at which the rate of enzyme activity is highest or maximum is called optimum temperature. You can write this slide information or previous slide information. Okay, if the temperature is raised still further, a temperature optimum is reached. Okay, after that reaction will be decreased. After certain point, however, an increase in temperature causes decrease in the reaction rate due to denaturation of the enzyme substrate and disruption of the active site. Okay, beyond optic beyond optimal temperature, what happens here, rate decreases. Once optimal temperature, highly reactive enzymes which reacts and rate of enzyme activity is done at the optimum temperature, then here you can see the decreasing curve. It means beyond optical sorry beyond optimum temperature, the rate decreases. Effect of pH. The rate of enzyme catalyzed reaction varies with pH as in the graph. Okay. The plot of pH, the against V naught gives a bell-shaped curve indicating the rate increases with slight increase in pH, then decreases. Okay. The decrease in the rate, which is the when uh, when pH is increased. Okay, when pH is increased or decreased due to the changes that occur in the ionic or polar groups at the active site. The large deviation in pH leads to denaturation of enzyme molecules. Okay, or there will be a disruption in their native configuration or three-dimensional configuration. The optimum pH of enzyme varies with their location. The optimum pH for the pepsin is 2, okay, pH 2, while that for alkaline phosphatase, that is 10 to 11. Okay, here you can see this is the pH 3, 6, 9, like that. You have a pH here, and here you have the velocity, rate of the chemical reaction or enzyme, we can write. So as and when increases pH, what happens here? Also, the enzyme substrate complex affinity will be increased. Once this particular uh, reaction which reaches to the optimum temperature and below the this, for example, here 6 is the optimum pH for this particular experiment. If that pH of the concentration are when increased in the graph, that time what happens, they can see the falling curve or decreased curve can be seen. Now further pH effects, that is tells us about the acidity and basicity of solution of pH scale 1 to 14. pH at which the activity of enzyme is highest is called optimum pH. Okay, change in pH leads to change in ionization of amino acids. Okay, that brings change in shape and structure of protein which disrupt the functions of proteins. Okay, at low at low and at very high temperature, changes the enzymes and substrate and bond may not form. Okay, if there is a high pH also, bond will not form. If there is a higher pH also, bond will not form, and that affects the rate of reaction. Okay, now further continuation, the example of effect of pH. Amino acid chains contain groups such as COOH, okay, and NH2 that readily gain or lose H plus ions. As the pH is lowered, that is acidic, an enzyme will tend to gain H plus ions. So the enzyme's shape is disrupted. Okay, whereas active site disturbed, and there won't be an ability to form bonds between the enzyme substrate complex. Okay, likewise, as the pH is raised to 11, 12, 13, 14, like that, no, what happen, they lose their structure. Okay, it means there also they will get disturbed. Okay, ability, they'll disturb the ability to form bonds between the enzyme and substrate. Many enzymes function properly at a neutral pH range and are denatured either at extremely high or low pH. Okay. To conclude here, you can write this information also. Okay, increase in the hydrogen ion concentration, that is pH, considerably influences the enzyme activity, and bell shaped curve is normally obtained. Okay, each enzyme has an optimum pH at which the velocity is maximum. Below and above this pH, the enzyme activity is much lower, and at extreme pH, the enzymes totally inactive. Most of the enzymes of higher organisms show optimum activity around neutral pH. Okay, 6 to 8. Therefore, however, many exceptions like pepsin, the acid phosphatase, and alkaline phosphatase, okay, enzymes from fungi and plants are most active in acidic pH. Okay, hydrogen ions influence the enzyme activity by altering ionic charges on the amino acids at the active site. Okay, and also can recall about the substrate, enzyme substrate complex, etc. Now let us understand aids, sorry, of this particular further effect of salt concentration. Okay, if the salt concentration is closer to zero, the changed amino acid side chains of the enzyme molecules will attract to each other. Okay, the enzyme will denature and form an inactive precipitate. Okay, if on the other hand, the salt concentration is too high, okay, normal interaction of changed groups will be blocked. New interactions will occur, and again the enzyme will precipitate. An intermediate salt concentration, such as that of the human blood, which of 9.9 percentage, or cytoplasm, is the optimum for most of the enzymes. Now let us go through effect of activators and inhibitors. Many molecules other than the substrate may interact with an enzyme. If such a molecule increases the rate of reaction, it is an activator. Or if it decreases the reaction rate, it is an inhibitor. Okay? These molecules can regulate how fast the enzymes act. Effect of product concentration. Okay, the accumulation of reaction products generally decreases the enzyme velocity, right? For certain enzymes, the products combine with the active site of enzyme and form a loose complex, thus inhibit the enzyme activity. Okay, in the living system, this type of inhibition is generally prevented by a quick removal of products formed. Okay, so this is all about the product concentration. Hope I made myself clear to you about the factors that influence the enzyme activity. In that, major one is enzyme concentration, substrate concentration, temperature, and pH. Okay, thank you everyone for watching this video. I look forward to see you in my next video. Happy learning. I, Dr. Pratibha K, signing out for today. Stay tuned with me. I'll be back with some more videos. Thank you everyone for watching this video.